So what is CRISPR-Cas9? CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeat. This name is referred to the unique organization of short partially palindromic repeated DNA sequence that found in the genome of bacteria and other organisms. CRISPR consists of two components, a non-specific CRISPR-associated endonuclease which is Cas9 and a guide RNA which also called as gRNA. The gRNA is a short synthetic RNA composed of a scaffold sequence necessary for Cas9 binding and a user-defined about 20 nucleotide spacer or targeting sequence which define the genomic target to be modified. What does CRISPR-Cas9 do? CRISPR-Cas9 is a part of a microorganism adaptive immune system. It protects bacteria from viral invasion by cutting off part of the viral genome. It basically works in 3 steps. Step 1. Adaptation. DNA from an invading virus is processed into short segments that are inserted into the CRISPR sequence as the new spacer. The spacer is a library of viral genome which had previously attacked the bacteria. Step 2. Production of CRISPR RNA. CRISPR repeat and spacer in the bacterial DNA undergo transcription. Transcription is the process of copying DNA into RNA. Unlike the double-chain helix structure of DNA, the resulting RNA is a single-chain molecule and it's referred to as gRNA. Step 3. Targeting. gRNA guide bacterial molecular machinery to destroy the viral material. Because gRNA sequences are copied from the viral DNA sequence during adaptation stage, they are exact match to the viral genome and thus serve as excellent guide. The figure had shown the step of CRISPR-mediated immunity. CRISPR are the region in the bacterial genome that help to defend against invading virus. These regions are composed of short DNA repeat and spacer. When a previously unseen virus infects a bacterium, a new spacer derived from the virus is incorporated among existing spacer. The CRISPR sequence is transcribed and processed to generate short CRISPR RNA molecule. The CRISPR RNA associate with and guide bacterial molecule machinery to a matching target sequence in the invading virus. The molecular machinery cut up and destroy the invading viral genome. CRISPR-Cas9 is first discovered in E. coli in 1918, while the function of CRISPR-Cas9 had verified in 2007. The scientist has first demonstrated that CRISPR-Cas9 can be used to edit purified DNA in 2012. Application of CRISPR-Cas9. Gene editing is one of the applications of CRISPR-Cas9. It removes the gene which translates to genetic disease, such as Huntington disease, or an impreferable gene. Gene editing is illegal in most of the country, but it is legal in USA and Australia.
Since the discovery of CRISPR-Cas9, scientists have been looking for ways to incorporate it into different science fields to benefit the population. Most focus on its potential in the medical fields. A recent study believes that CRISPR-Cas9 is able to inhibit cancer growth. This study uses virally driven cancers, such as cervical cancer induced by HPV, as its model. HPV-16 and HPV-18 cancer cells rely on E6 and E7 proteins to survive. When genes coding for expression of E6 and E7 proteins are removed, it resulted in cell death. In laboratory mice, knockout of E6 and E7 genes resulted in the inhibition of tumor growth. Aside from targeting E6 and E7 genes, CRISPR-Cas9 can also be used to target other specific regions of a genome. One example would be by targeting the gene which blocks the expression of viral capsid proteins, which are highly immunogenic. Hence, by allowing expression of viral capsid proteins, the host's immune system can be triggered and will eventually lead to the annihilation of the virus.
Created using Powtoon.